We're about to move on here, but it just occurred to me as I was about to start this, the last segment of the lecture. I emphasized here that a cellular protease is used to cleave GP116 to GP120 and GP41. And if that cleavage doesn't occur, then the virus isn't infectious. And that's absolutely true and really important. I just wanted to also mention that other preproteins of the virus. So, for example, the GAG and the POL genes, they're translated as one polyprotein, and they are cleaved by the HIV, the viral protease. Right? So, protease is important for cleaving GAG and POL. That's the viral protease. But for cleaving the glycoprotein, GP120 and GP41, you need the cellular protease here. We know that AIDS is due to the slow depletion of the helper T cells in your immune system, right? So your immune system just gets slowly demolished over time. You lose your helper T cell function, and that's what leads to AIDS. But some other pathology that can come with HIV. So, like a lot of other enveloped viruses, if you have the expression of the glycoprotein on the surface of the cell, this can lead to fusion with neighboring cells, and then you get your multinucleated syncytia. So we've talked about this phenomenon with other viruses. You can get the same thing happening with HIV, and it can lead to cell death. We mentioned previously, on the last couple slides of part 3 here, that we've got that viral protein R, the VPR. And VPR can lead to apoptosis. VPR can cross the blood-brain barrier. VPR permeabilizes cells to cations. Some HIV, as it evolves over time, can also become neurotropic, can infect nerve cells. And so for some patients with AIDS, they get AIDS dementia complex, where we've got either damage from the virions or from VPR that alter the immune response, cause inflammation, lead to death and damage of nerve cells, and this leads to these symptoms. So impairment of reasoning, behavior, and changes in the ability to move. So this happens about 20% of the time with AIDS patients. So initially during infection, the viruses that can infect your macrophages are usually the initial viruses that infect you. And after a few days or weeks of infection, the virus kind of switches tropism, and it prefers to use a different chemokine receptor. So over a period of time, it prefers to use CXCR4. However, the initial infection is with CCR5. And so if you have a different version of CCR5, where there's a deletion here at residue 32, if you have a mutation or an altered CCR5 and you can avoid this initial infection, then you are genetically resistant to HIV. So there are a very small percentage of people that have this mutation, and so HIV during early infection can't infect their macrophages, and so then they're resistant to HIV. And there's a hypothesis that this mutation became more prevalent in the population because this mutation also confers resistance to smallpox. And so people that survived smallpox were more likely to have this mutation. So the ones that survived smallpox continued to reproduce, and they passed this mutation on. So there's this theory that people who were resistant to smallpox selected for this mutation in the population. So you have a small percentage of the people nowadays with this mutation in CCR5 that are resistant to HIV. Now, how do we treat HIV? Well, as I mentioned before, we've got antiretroviral therapy. This also is sometimes abbreviated as highly active antiretroviral therapy. HAART, I don't know, I guess people thought it sounded cool. So you can be treated with HAART or ART. It used to be, so this is a old recommendation, that when your T cell count dropped to less than 200 cells per microliter, should have units there, that's when you should be started on ART. Now they've changed this, and so now they recommend immediate treatment. The very first drug that was approved was AZT. This is a nucleotide analog, a thymine analog. So it looks like thymine, but it's not. We've talked about nucleotide analogs previously. And so this would cause a problem with reverse transcriptase. However, drug resistance emerged, as you would expect, right? So now what they do is they combine at least 3 different antiviral drugs. And that's where you get the ART or HAART. And it's expected that the virus, in any one cell, the virus will replicate and you'll get a mutation and it will be resistant to one of these 3, but not all 3. And so you'll still have those 2 other drugs that will kill the virus. And so using a cocktail ensures that even if the virus picks up one resistance mutation, it's not going to become resistant to the entire cocktail all at one time. That would be statistically exceedingly unlikely, like statistically impossible. I mean, I guess in theory it's possible, but exceedingly unlikely that you could develop resistance to the whole cocktail of mutations, or a whole cocktail of drugs at one time. So we've got several different targets for our antiviral drugs. We can block entry. We can block reverse transcription. We can block the protease. So remember I mentioned the cellular protease is going to cleave the glycoprotein, but the viral protease can be targeted, and we can target the viral protease that's going to cleave the GAG and the POL genes. You don't want to target the cellular protease because that would also screw up other cellular functions, but you can target the viral protease. We can target fusion. We can target binding to CCR5. We can target integration. So there's lots of different ways that we can target the virus, and by using a cocktail of drugs that have different targets, we can avoid the virus developing resistance. The problem with this is that it costs money, right? I hate it when people say money isn't everything. Yeah, well, you've never been poor. Money is really important, especially when you don't have any. And medication costs money, and a cocktail of medication costs money. And if you have insurance, that's great, and insurance can cover this, but there's still a lot of people in America that don't have insurance, and there's people all over the world that don't have adequate access to healthcare and treatment. So now one of the biggest pushes is trying to get everybody who needs this therapy the medication that they need. One of the biggest problems with HIV, right, is that you can be sick for decades and not know that you have HIV. And you don't have to be a particularly promiscuous person. Let's say you have one new partner a year. For 10 years, you've spread it to 10 people. And you're not trying to spread it. You don't know that you have it, right? If we could get better access to testing and better access to treatment, we could drastically slow down the spread of this virus. It's that being asymptomatic and still being able to spread it is such a big problem. Kind of similar to the coronavirus right now, right? Is that it's really infectious and a lot of people get it, but they're asymptomatic, but they can still spread it to other people. All right, so what about a vaccine? Well, that's really, really hard. And one of the biggest reasons is the virus evolves so quickly. So the reverse transcriptase has no proofreading. So that means it accumulates mutations really, really quickly. And we already kind of went over how there's different strains of the virus all over the world. And when you get infected, you actually get infected. So say you have, say you're at this point, right? And each one of these different colors is a different variation of the virus, because as it's replicating in you, it's accumulating all of these mutations. These mutations help it avoid your immune system, and you've got all of these different variations of the virus. When you sleep with somebody, or when you share a needle with somebody, you're introducing all of these different viruses to that other person, but not all of them are really going to be good at infecting. Maybe only the blue ones, let's just pretend, maybe only these blue ones are actually super infectious. Maybe the other ones are well adapted to your immune system, and only these blue ones are going to be good at infecting macrophages, right, in the new person. So these blue ones infect the new person, and it replicates in the macrophages, and then it switches, and it starts replicating in the T cells, and then it starts evolving really quickly because there's no proofreading, and now you have this rainbow of viruses again, right? And so the actual genetic diversity with the virus in one person can be greater than the diversity between multiple people, or the diversity between like people from different parts of the world. There's tremendous variation within just one person of the virus. And trying to find a vaccine that can neutralize all of those viruses is going to be exceedingly different. So, is it theoretically possible? Yes, because we know that there are elite suppressors, we know, or non-progressors. So we know it's possible to develop broadly neutralizing antibodies broadly neutralizing antibodies that can neutralize all of these different variants. But getting people to produce these broadly neutralizing antibodies in response to a vaccine is exceedingly challenging. And I really hope that one day we get there. But until then, we really need to focus on lowering the cost of treatments. So we're almost done with the semester. There's one more lecture. It's on the prions and virions. I promise that'll be really short. So do the best you can. Email me if you have questions, or talk to me in Canvas chat. And I hope you guys are doing well.